So I want to do a little bit of an overview here before we move on to some regulation aspects of how cells regulate what they're going to do when, and then before we go on to talk about electron transport chain. So this particular picture that we're looking at is not the cell, this is the mitochondrion. So you might label out here is the cytosol, out here is where glycolysis was going on. And glycolysis produced pyruvate. Pyruvate has to move into the mitochondrial matrix, which is this compartment. You might add that on here too. Pyruvate has to move into the matrix. It's acted on by pyruvate dehydrogenase in here to make acetyl-CoA in the matrix. And acetyl-CoA donates those two carbons into the citric acid cycle. They combine with oxaloacetate, they make citric acid. And then during that cycle, there's CO2 released, there's some more NADH made, and there's some GTPs made. So I think I want to add something onto here as far as numbers and overview. So out here we have glycolysis, and if we're talking about per glucose, we had 2 pyruvates produced during glycolysis. And we also have 2 ATPs out here from glycolysis, and we had 2 NADHs also from glycolysis. There is a CO2 release step right there that's not shown. During citric acid cycle, there are 4 CO2s released. There are 6 NADH made. Also a couple of GTPs, also a couple of FADH2s in the matrix. So we're going to be focusing on, when we get to the electron transport chain, this group. Maybe that box a little bit bigger. This group of stuff right there. The electron carriers, NADH and FADH2. GTPs, it's in the box, but I didn't mean for it to be. That's what we're left with. So at this point, these electron carriers, they are holding most of the energy from glucose that remains. They're holding that electron energy, carrying it around with them into the electron transport chain. The other thing I want to remind you of, or maybe introduce for the first time, is that all the ATPs and GTPs that we've made up until this point were made by substrate level phosphorylation. As opposed to oxidative phosphorylation that the electron transport chain does. So I think that you've probably heard these terms. I think that they're weirdly named and hard to understand. So here's how I understand them. Things that are made through oxidative phosphorylation, that's using ATP synthase. So that spinning turbine-looking protein that lets protons flow through and it magically turns that energy into ATP, that's oxidative phosphorylation. And it's driven by proton gradients, really. If ATPs are made some other way besides that, that's substrate level. So substrate level phosphorylation is kind of like individually moving phosphate from one molecule to another. That's substrate level. Substrate level is a slow and wimpy way of making ATP. It's made 2 ATPs through glycolysis, 2 GTPs in the citric acid cycle. But electron transport chains that are coupled with ATP synthases, they're making ATP like gangbusters through a completely different way. So not taking a phosphate group off of another molecule it's stuck onto, like substrate level, but using ATP synthase to join these two together quickly. That's oxidative phosphorylation. So organisms that can do this have an ATP advantage, and they have a life complexity advantage over organisms that only do substrate level. That substrate level phosphorylation and oxidative phosphorylation, those are really prevalent on standardized tests for some reason. It seems a little overemphasized to me, but good terms to know. So remember that from standpoint of electron transport chain, NADH and FADH2 are the important incoming players there. Now, this picture is scary, but it's also really cool. And this is sort of relating back to this slide here. So the concept I introduced here that, these cycles are useful for more than just making ATP eventually to produce intermediates. Expanding on that gives us this kind of picture. And this looks like just a lot of random dots and lines, but this is a depiction of the major biochemical pathways that intersect with glycolysis. So glycolysis would be the red here to here. Here's that in-between step. Here's citric acid cycle. And every line and every dot on there is a biochemical enzymatic process that has been worked out. It's not made up. It's like carefully constructed. A ton of work. One thing I love about this is that little dog leg. What is that? You don't have to remember the name specifically yet, but what is that little detour?
That's where when I forgot what it was, I can't remember the name, but when it breaks apart, you got one molecule that was ready to go, and the other one had to be made like that.
Yeah, this is dihydroxyacetone phosphate. And so, like, I don't know, sometimes recognizing something little like that in a large mess, it's like, oh, I know what that is. I know what that is. That's a citric acid cycle. But what I want to point out with this is every place where there's an intersection, like here, glucose 6-phosphate, we talked about that can be a building block for nucleotides. So every place there's an intersection, the cell has a decision to make. Am I gonna put more into glycolysis, or am I going to take more out to build nucleotides?
Pyruvate has a lot.
Pyruvate has a lot.
So, well, why would pyruvate be able to be used in so many different things if it's like energy is just so important for processes overall? I feel like
So
once you got to
things.
that point.
Yeah. So the question is, all right, pyruvate is used for a lot, which it is. Is that crazy? It's not crazy. It is what it is. It's a building block for a lot of things. So it's a versatile 3-carbon molecule. If you're going to remember how to draw one thing out of this, it should be pyruvate. For tests, standardized tests and stuff. Pyruvate. So somebody was asking, why would the cell, if pyruvate is so important for energy, why does it use it for so many other things? Well, the other things are important too. Like, you could store up energy all day long, but the reason to make the energy is to use it to build things, and pyruvate is used for both making energy and to build things. The cell keeps a lot of it around.
It just seems like it goes, like, the cell goes through a lot of steps to get to pyruvate, and then, like, all the ones before it aren't used for as much as, like, that one is. So it's just surprising to me that it would do so much work just to get to one thing, and then like, that one thing is used for everything.
Instead of using glucose for everything.
Yeah.
Some of it just has to do with, I guess, from an entropy standpoint, the reason that cells are using glucose is because plants are building 6-carbon sugars, and that's the major form available in the world. So getting a 6-carbon sugar down to a 3, this is like Legos. Like, you could build almost anything out of, with Legos it's those little 2 blocks probably. If you had enough of those, you could build almost anything. If you've only got 6s, it's less versatile, so.
Yeah, I see.
But the cell keeps lots of pyruvate around. Like, the pyruvate pool is bigger than other pools, more populated than other pools. It's hugely important, intermediate. So the cell, it's almost miraculous to me. It can monitor the levels of all of these things. There's no brain in the cell. A lot of these molecules are floating around in combined pools in the cytoplasm, but the cell can monitor levels, and it can make adjustments to which processes to promote and which processes to suppress. So, amazingly, there have been some years when I didn't bring up the term pool until this day. You know a lot about it this year. Cells know, they know how much of each dot they've got on there. It's amazing. They know that. And they will adjust which process is dominant depending on what they most need. And we're going to look at just a brief example with glucose. So glucose, here's glucose. It seems like it has not enough connections coming off of it, but the cell is monitoring how much glucose it has. And glucose is quite important for feeding into all of these other pathways. So this stuff is up in here, but if you look, it's mostly connecting down here, and glucose is starting material for that. So keeping glucose supplies constant is a big deal for organisms. If organisms detect that they have a lot of glucose, like really a lot, they will do something smart and store it as glycogen. Where do we store glycogen? The liver, and then skeletal muscle. For later. I might need this glucose later if I have to take like an immediate extreme action. So build glycogen out of excess glucose and store it up in the body. That's what people do. Other organisms do different mechanisms of storage, but that's the response to having a lot of glucose. If you have a lot of glucose and you have a lot of glycogen, and you don't even have any more room for glycogen, glucose can be processed somewhat through these pathways and stored as fat. So that would be secondary storage. Dalton's going to be somewhat talking about this stuff in his seminar. Like, not about glucose and glycogen, but generally like, oh, I've got so much I can store fat. Lucky me. Is that on?
Yeah.
Okay. I mean, I want it to be on. Okay. What if you have the opposite problem and you don't have enough glucose? So if blood glucose is low, where do we turn first? What'd you say?
Fatty acids.
Fatty acids from where?
Long chain fatty acids.
We go to glycogen first, if we've got it. Then we go to fatty acids. Then, like, long-chain fatty acids. But while that is going on, even if you're burning fatty acids, you're actually building some of them, you're actually building some of them back into glucose, like for your brain, and tissues that are picky about what fuels they use. So if you don't have enough glucose, cells can build it. They can run some of these pathways backward and build glucose, and they do so. So when we looked at glycolysis and some of the arrows were reversible, some of those enzymes were discovered and named because the researchers were focusing on the building process, not the breaking down process. So if I remember right, fatty acids feed into acetyl groups and then can run backward to build glucose so that we can supply the brain and so on. That process of making glucose is called gluconeo, sugar new making, gluconeogenesis. And that's a really good word to know. Gluconeogenesis. It is the opposite of glycolysis. So, sugar break, sugar new make. None of those words. I really like it when words make sense to what they mean. I think that's all I'm going to say about that slide. This picture, I just wanted to come back to it, and these look intimidating when you first look at them. I remember some professor in my college had this up on their wall and with all the names filled in and stuff. It's like, who could care about that? And theirs was probably the plant map too. Like, who could care about that? It's really important, and it's really beautiful too, that our cells have the capability to do this. And that's not even all of it, that's just some of it. I want to go ahead, since we've got a decent amount of time and a test in a week, I want to go ahead and get started a little bit on the electron transport chain. So if you would stop the video again, we'll have 3 short videos today.